Hello, my name is Andres Sorta, and today I'll be talking about the mitochondria and its structure. So, the mitochondria have many functions in the cell, and it is also the cell's main source of energy in the form of adenosine triphosphate, also known as ATP. So, the mitochondria power the processes that occur and take place within the cell and the organism. This is because the mitochondria is the site of oxidative phosphorylation. Oxidative phosphorylation accounts for most ATP synthesis by non-photosynthetic organisms. So if we look at this diagram, we see that the mitochondrion consists of two phospholipid bilayer membranes. The two membranes are the outer membrane and the inner membrane. So in the diagram, the outer membrane would be out here, and the inner membrane would be here. So the outer membrane consists of these proteins called porins. And these proteins allow facilitative diffusion of ions and small molecules across the permeable outer membrane. Now, in between the outer membrane and the inner membrane, we find the intermembrane space. Now, we go ahead and look at the inner membrane. We see these convolutions, which are called cristae. Now, these occur in order to maximize the surface area of the inner membrane. So the reason we want to maximize this area is because there are proteins, transmembrane proteins, that have to go here. So for example, a liver mitochondrion may have more than 10,000 sets of electron transfer systems. These systems consist of many proteins, and without these folds, we would see that these proteins would simply just not fit. So now, taking a closer look at the outer membrane and the inner membrane, we can see some of the proteins that I mentioned before and how they're involved. So we find that the outer membrane is readily permeable. So this means that small molecules and ions are able to flow through easily. The phospholipid bilayer contains these large integral proteins called porins. And these porins allow molecules like NADH and pyruvate to flow through the bilayer into the inner membrane space. So what we will find here is that the inner membrane space and the cytosol will actually have equal concentrations of ions and small molecules, and this is due to the easy flow of these small molecules and ions through the porins. So now let's take a deeper look at this porin protein and how it works. So I've pulled up a 3D image of it. This is a 3D structure. This is a very specific porin protein, but all of them have a similar structure as they contain similar functions. So what we see is the creation of this pore by these beta strands with random coils allowing for turns, and repeats until you basically get this circle through the outer membrane. So what we will find is the outer part of the pore to be hydrophobic in order to match the inside of the phospholipid bilayer, while the inside of the pore would be hydrophilic in order to match the conditions of the inner membrane space and the cytosol. So now we have basically created a hole through the outer membrane that allows a flow of molecules that are small enough to fit through here and ions. Again, since we have the flow of small molecules and ions through the outer membrane, we are able to ensure that the inner membrane space and the cytosol have similar concentrations of ions and small molecules. Where concentrations differ is larger proteins that cannot get through this porin to get out into the cytosol or into the inner membrane space. So now, considering these NADH molecules and pyruvate molecules from glycolysis, we have been able to get them through the porin into the inner membrane space, and now are left at a roadblock, as the inner membrane is not easily permeable the way that the outer membrane is. And now let's go ahead and talk about some of the ways that these molecules are able to get into the matrix of the mitochondria where they will be used for the synthesis of ATP. So now that we have established that NADH and pyruvate can easily get into the inner membrane space, let's go ahead and talk about how they are able to get into the matrix. So as we can see here, we have the inner membrane space and the matrix. So since the inner membrane is impermeable, we have to use these integral proteins, and they can carry these molecules from the inner membrane into the matrix. Here in the inner membrane, we will find a shuttle proteins and these complexes from the electron transport chain. So we have shuttle molecules like the glyceryl 3-phosphate that transfer the electrons from NADH into ubiquinone, reducing it to ubiquinone, where it takes part of the ETC by going directly into complex 3. So basically, the NADH molecules that come from glycolysis will go into a shuttle where they will be turned into ubiquinol molecules that will go on to the complex 3 to form ATP. So the matrix contains most proteins found in the mitochondria, and many of these proteins are part of cycles that occur within the matrix. So as we can see here, we have all the proteins needed for the pyruvate dehydrogenase complex are able to come together inside the matrix to turn pyruvate into acetyl-CoA. Then the pyruvate from glycolysis that is now acetyl-CoA can go on to the Krebs cycle or the citric acid cycle, where it is able to generate these NADH molecules. And also, we are able to get NADH molecules from the beta oxidation pathways and the amino acid oxidation pathways. So now that we have all this NADH from these multiple pathways that are occurring again in the matrix, the NADH will enter the ETC through complex 1, therefore going on to create ATP in the same way. When the NADH molecules enter the ETC through complex 1, they will create 2.5 molecules of ATP per molecule of NADH. However, when NADH comes from glycolysis and straight into complex 3 through this shuttle protein, we see that it only creates 1.5 molecules of ATP, and this is due to the lack of the extra protons that the complex 1 and 2 would be pumping. So since we have those less protons, we will have less ATP formation from NADHs that come from outside of the mitochondria and from glycolysis. So now when talking about the structure of the mitochondria, we see that from the ETC, electron transport chain, we create an electrochemical gradient across the inner membrane. So since protons are being pumped through each complex out of the matrix into the inner membrane, we will find that we create these set of protons in the inner membrane while keeping electrons in the matrix. So this creates, again, a gradient going across from these protons. So that is all for the mitochondrial structure, and thank you for watching.